So this was how we got to our short list with different AKRs and ADHs, and going back to some other concepts that we discussed earlier in the course. Two of these, from each category of superfamily, DKGA and AKR and YQHD and ADH, were actually within one genetic operon with this YQHC transcriptional activator. And that was of heightened interest to us, because this is exactly the kind of setup you would expect if, just like we talked about biosensing, if there's some kind of response to stress or to the presence of an aldehyde directly, you would expect some of these different enzymes that are capable of doing this reduction that's supposed to help the cell. You'd expect them to be clustered together and maybe governed by the same kind of response. And so I don't think I'm going to show it in this presentation, but later when we actually did an aldehyde challenge, we did that with benzaldehyde sort of retrospectively on our short list rather than doing it at large. We could have gone that approach, but you definitely see a surge in the expression level of these proteins. And not only these, at least one or two of the other ones really rise in expression level in the presence of an aldehyde. Okay, so to actually test these different knockouts, we had this setup, as I mentioned before. We transform a strain with the gene encoding a CAR. We would add our acid substrate, in this case, to tubes that were capped with a high degree of headspace at an angle so that you'd still get some good mass transfer and keep maintenance of aerobic conditions. But we wanted to keep the vials closed because some of these molecules are pretty volatile, like your benzaldehyde in particular, even the benzyl alcohol. And then going back to some of the frameworks that we discussed for measurement and for analytics, in this case, we would use simple HPLC of the culture media. So we would spin down cells, take just the liquid, and this approach doesn't always work, depends on your chemistry. We would find that actually, accounting for some loss to the headspace, but if we were to take early time points in particular, our mass balance would close just in the free liquid. So we would see both formation of these kinds of downstream products, and we'd see them coming back out into the cellular media. So we could then safely assume that our concentrations were equilibrating reasonably well. And so then with just HPLC based on the peak areas, we could do quantification. And so I'm only going to take you through a few results here, just to, the sort of minimum results required to make the point, and then not to belabor the point. But what we were able to do is create different subsets of deletion strains. So one approach would be, hey, we have these 6 targets, let's just delete them all. And as we talked about earlier in the genome engineering tools section, the ability to multiplex and investigate things combinatorially, well, that can be really helpful. That can be helpful, one, if you have an RBS library and you've got 20 different targets in the genome like you had for lycopene biosynthesis, and then you don't know what combination you want. Well, this was another situation. This was just deletions, but we didn't know what set of deletions we really needed. And you could err on the side of just making more. But similarly with the lycopene MAGE example, you might end up in a situation where now you've made more knockouts than you needed to, and maybe one of them is really going to hurt the cell in terms of it might have some other action that it performs because of glyoxylate or methylglyoxyl building up in the cell. And so you only want to make as many interventions as you have to. There's some trade-off there. And so we could look at that here. We're grouping, this is a table that just shows the different genes in rows, and I've separated the AKRs and the ADHs. And so we can knock out a couple from each category. We can take out just the operon that I talked about. We can take out the operon plus all the AKRs, operon plus all the ADHs, or the whole combination in our shortlist. And as we were doing this, we had no idea whether the shortlist would be good enough. And, for me, it was maybe, mentally thinking that this could go on for a long time. Like with any kind of real novel research effort, you don't know that it's going to work. And maybe most of the time it actually doesn't. So we were very lucky in some sense that we could do these knockouts and then do the measurement where we look at concentration 24 hours after benzoate addition. And we would start with 5 millimolar benzoate, a reasonable amount to be adding to cells. And we had this MG strain is sort of this wild type E. coli. And then that's the ancestral strain that we're making all of these knockouts to. And what we saw is, okay, you can make these knockouts. You still end up, here are three compounds. You still end up mostly almost exclusively with benzyl alcohol. And again, it doesn't get you back to 5 because there's some loss into the headspace, and given the volatility of these compounds. But you don't see any benzoate anymore, so went all the way. You don't see any benzaldehyde. That's too bad. That's what we want. Maybe you see a little here. That's within error of 0 though, so probably nothing there. You don't see any here. Oh, but wow, something changes quite a bit once you've knocked out the operon and the other ADHs. And so right away with this subset, we seemed to think that the ADHs have a greater role to play in creating this byproduct formation than these AKRs. And then, so you can compare some bars here, but this has more to do with the kind of variability that you get from within supplementation experiments, especially based on when you induce your CAR expression and what amount of CAR you get. That has a lot to do with how quickly your benzoate goes down and forms benzaldehyde. Because of that, the real key indicator that we wanted to compare is how much benzyl alcohol do we really have, and it's pretty comparable in these two strands, essentially telling us that even though we landed on this final strain to just have the other knockouts just in case, and there are some situations where you do want these AKRs to be knocked out. I guess I'll just mention right now, if you had glucose provided to the cell and you were trying to make aldehydes from glucose as a carbon source, we found that actually these two strains do perform differently in that case. Likely meaning that these AKRs, which you didn't have to knock out in this experiment, do get turned on, the gene regulatory influence there. So in this case, we liked this final strain. It had a decreased or reduced ability to perform reduction on aromatic aldehydes, and so we called it the rare strain for reduced aromatic aldehyde reduction. And 2 quick points that I'll make before I transition onto our next topic is that this we started to see in the study that we first looked at this, that this could have applicability to more than one aldehyde. So we focused first on aromatics, and that was really the title of our first paper on this topic. And part of the reason for that is because the system works really well for aromatic aldehydes, at least in particular the ones we tested, also others. So if you look now at a similar experiment, but we added vanillate instead of benzoate, you can look at the concentration 48 hours after addition of vanillate, starting with, again, the 5 millimolar. And there are a few things that are different here. We picked 48 hours, so 2 days rather than 1, because the carboxylic acid reductase works slower on vanillate. So the amount of vanillin that we're getting made in the cell is the rate of its formation is decreased. And we also thought that, maybe these AKRs or ADHs are also going to act slower on vanillin. And so indeed, when you look at the wild type strain after 48 hours, you do still see some vanillin is around, but you can clearly see that most of it has converted to vanilla alcohol. And here, because of the reduced volatility, you get a nice clean millimolar, 4 millimolar, you get back your 5. Then when you look at the rare strain that we engineered, you see this really nice concentration profile where you have no detectable vanilla alcohol, even after 2 days. You've just completely abolished formation of this product, at least with vanillate addition. And so this is meant to illustrate a couple of things. One, that this is a clear way in which one might need to do genome engineering in order to build up the product that you actually want, and how genome engineering can take us to new frontiers where it's not necessarily the case that we could use any kind of predictive software. And in fact, you know, we made this point that something like OpNoc wouldn't tell you that you needed to do this. It both wouldn't tell you that vanillin would get converted to vanilla alcohol, and it wouldn't tell you which genes do that and what you'd have to knock out. So this is still in that phase of experimental discovery work. The other thing that this strain was then subsequently used for in a variety of ways is that if we go back to those tables where you looked at acetaldehyde, hexanal, methylglyoxal, these other aldehydes, it turns out this strain, once you make these knockouts, really just these 6 or 7 genes if you include the transcriptional activator, which was partly included as a control, you know, you find that if you're biosynthesizing the aldehyde that might encode or that might connote or convey a apple or other kinds of flavors, those aliphatic aldehydes, you get increased accumulation of those too. You don't get, like in this case, the complete prevention of alcohol formation, but you get a substantial boost in the amount of aldehyde you can make. In other words, you substantially decrease the rate of alcohol, of aldehyde reduction. You don't abolish it, but you minimize it. And so those approaches can be really valuable at the end of the day, not just for making aldehydes, but for making anything that might come from an aldehyde. And so looking forward in the field, in metabolic engineering, there's still a number of ways, I would say relatively low-hanging fruit opportunities for genome engineers to come in and be able to say, well, these are products or type classes of products that we couldn't make before. The cell has something that we don't want. And this general idea of competition, that your cell is not a blank canvas. It would be like the chemical engineering analogy would be building a chemical plant where there's already one, almost retrofitting, except you don't know what the current chemical plant does completely, or there are some unit operations that nobody has figured out what they do. And so you can imagine how if you were a chemical engineer trying to do that, that would be a frightening endeavor. But that's essentially still a lot of what we're doing here with the cell.